Good morning, everybody. Hope you can hear me well. So it's my great pleasure to open this second and last day of the BraYn edition, the third edition of BraYn to 2020. So my name is Rosa Paulicelli, and together with Enrica Boda and Sonia Calabrese, we will share this first session in neurophysiology and neuroplasticity. So before we start, I just would like to give you a few reminders. First of all, that all of you will be invited to express your preference at the end of all your presentation for your favorite one, because for this audience award, because the winner will have access the free registration for next year, the fourth edition of BraYn. Another thing I need to remind you is that all the questions will be asked at the end of the session. So please, don't wait until the last minute. You can start typing your question in the Q&A chat box that you see on your screen, and we will read them for you. But don't forget to introduce yourself and also to write to specify the name of the speakers to whom you would like to address your question. Also, I'd like to remind to all the presenters, please, to keep the time. This will allow us to have some nice discussion at the end of the session. Now we can move to the second speaker, Ayesh Jacob from Scuola Normale Superiore, Laboratory of Biology, Pisa, Italy, who will talk about Synactive, a genetic toolbox to study the proteome and connectome of learning and memory-associated synapses. Ayesh, you can share your screen.
So, good morning, everyone. I'm Ajesh Jacob, PhD student at Scuola Normale Superiore, Pisa. Today, I will be talking on Synactive, a genetic toolbox developed in our lab to study the proteome and connectome of learning and memory-associated synapses. Memory is known to be stored in the engrams. Engrams are neurons that are active during learning, which are necessary and sufficient for the memory recall. Current techniques allow us to study the engram at the cellular level. In our lab, we have developed Synactive to study the engram at the synaptic level. Basically, in Synactive, a gene, say GFP, is expressed under the control of 5' and 3' RQTRs. This makes the translation of the protein activity dependent. Once the protein is produced, they are enriched at the synapses with the help of PSD binding peptide attached to the C-terminus of the GFP. And the whole cassette is temporarily controlled with the tetracycline dependent promoter. We have used this construct to study the potentiated synapses in the mouse hippocampi after novel context exposure. In my talk, I will be explaining how to extend the Synactive to study the proteome and connectome of potentiated synapses. Let's begin with the proteome. In 2009, Fernandez et al. Has expressed PSD95 tagged with flag in the mouse brain, and they have used it to immunoprecipitate the synaptic proteins. We used a similar approach and made construct to express PSD95 flag in all synapses. So this will allow us to immunoprecipitate all synapse proteome. This concept will be referred as HSYN. And to make this activity dependent, we added the 5' and 3' RQTRs to PSD95 flag. This way, we will be able to express PSD95 flag only in the potentiated synapses, which will allow us to immunoprecipitate potentiated synapse proteome. This construct will be referred as ESAIR. We have made virus for both the construct and injected in the mouse hippocampi. Then the mouse receiving the contested fear conditioning were undergone, the mouse receiving the ESAIR construct were undergone contested fear conditioning, and the mice received the HSYN remained in the home cage. After the behavior, the brain was harvested and proteins were immunoprecipitated. The samples were then analyzed using the Western blot and by mass spectrometry. Immunostaining the mouse brain injected with the HSYN and ESAIR, we saw PSD flag expression. In the case of HSYN, we saw many PSD flag punctures, whereas in ESAIR, the puncture number was less. This is in agreement with what I explained before, because in the HSYN, PSD flag is expressed in all synapses, and ESAIR, it is expressed only in the potentiated synapses. We were able to immunoprecipitate PSD flag in both ESAIR and HSYN conditions. In agreement with the images that I showed before, PSD flag was expressed less in the ESAIR. Then we analyzed for the potential then we analyzed the non-PST interacting proteins in the samples, and we were able to identify BlueN2AB, ARC, S6 are enriched in the ECR samples. Then to get a molecular fingerprint of potentiated synapses, we did the mass spectrometry and did the analysis, and we were able to identify 93 proteins that are exclusively expressed in the potentiated synapses, and 122 proteins that were absent in them, and 690 proteins that were expressed in both samples but had differential expression. These are preliminary results, and we are further analyzing the data. Now moving on to the connectome. There is a structured connectivity between brain areas. But what about the connectivity at the synaptic level? GRASP is a tool that allows us to study this. Basically, in GRASP, half of the GFP is expressed from the presynaptic terminal, and the other half is expressed from the postsynaptic terminal. By itself, they are not fluorescent, but when they come closer, as in the case of the synapse, they become fluorescent. We used this approach and made some construct to express the presynaptic GFP, half presynaptic split GFP, and postsynaptic split GFP, and they are under the control of tetracycline promoter. So we made construct to express RTTA and TT tomato in the presynaptic neuron, and RTTA and synaptic neuron. If the system works, we will see presynaptic neuron in red, postsynaptic neuron in blue, and their synapses in green. This will tell if the two neurons are structurally connected, but nothing about whether they are functionally active. To make it activity dependent, we added the 5' and the 3' UTRs. With this, you will see GFP only when they are structurally connected and functionally active, not when they are just structurally connected. And we have also added a red fluorophore along with the post split GFP as an internal control. We validated the construct in the in vitro. We translated neurons with the pre-GRASP and post-GRASP. Then LTP was induced in the culture. Then they were fixed and imaged. In the case of normal GRASP, most of the synapses between the presynaptic and the postsynaptic neuron were expressing GFP. But in the case of synaptic GRASP, there were some synapses expressing GFP, but some synapses failed to express GFP. Then we modulated the activity in the cultures. As in the case of normal GRASP, there was no significant difference between groups for the GFP positive synapses. But in the case of synaptic GRASP, with the inducing LTP in the cultures, we could see an increase in GFP positive synapses. And the number of GFP positive synapses were reduced when we inhibited the potentiation using the NMDA blocker AP5. Then to check whether the GFP positive punctures actually co-localize with the synaptic proteins, we stained the neurons with the postsynaptic protein PSD95, and we saw an overlap in both GRASP and inactive conditions. And in both cases, they have a close opposition with the presynaptic protein synaptophysin. And most of the cases, GRASP signal was observed at the spine head. Then to check whether the synaptic GRASP positive spines are actually potentiated, so we stained the neurons with GluA1, and we could see an increase in receptor enrichment in the case of synaptic GRASP, and those spines were actually increased in the spine area. Whereas when we immunostained the NMDA receptor BlueN2AB, there was no significant difference in the enrichment values in both between the both groups. But in the case of synaptic GRASP, they had a higher spine area. To conclude, synaptic GRASP expression is activity dependent, and they co-localize with the synaptic proteins. And synaptic GRASP is expressed in the potentiated synapses because they have increased spine area and enrichment in the AMPA receptors. Currently, we are extending the synaptic GRASP to study the spatial distribution of potentiated synapse after the learning and the encoding phase in the CA3, CA1 connection and the DG and the internal cortex dentate gyrus connections. And we are planning to apply synaptic graphs to Alzheimer's mice models to study the synaptic dysfunction. With this, I acknowledge all my lab members and professors and the collaborators and the institutes for the funding and support. Thank you.
Hi all. It's time for starting the BraYn Educational Symposium session. And the next speaker is Chloé Dominici from the IBDM in Marseille with the talk titled 3D Analysis of Neural Network Remodeling in Pancreatic Cancer, sponsored by Miltenyi Biotech. So let's start. And Chloé, okay, it's
Hello.
your time.
Okay. Okay. Hello to everyone. So I'd like to thanks for the organizer of the conference and Miltenyi Biotech that gave me the opportunity to present my work that I did during my postdoc in Fannyman's lab in Marseille. And so I worked on the link between the neuronal network and the cancer cells. And in fact, Fannyman's team is initially a team that is interested in axon guidance. So they look how the neuronal network is properly achieved during the development. And for that, they study axon guidance molecules that could either attract or repulse the growth cones in order that axon can reach the right target tissue. But during the past few years, they are interested in axonal plasticity, not in a context of development, but in a pathological context, such the cancer. And interestingly, such an interaction, so with axons and tumors, has been already shown 70 years ago with Elmer Bucker and Rita Levi-Montalcini. And in fact, they were seeking some new molecules that could make axons growth. And so they grafted a tumor in chick embryo. And they found that tumor were able to attract nerves, and some newly axons were formed inside the tumor. So here we are again in an embryonic context where the nerves are very plastic. So could we find the same innervations in human adult cancer? And the answer is yes, because it has been shown, so with several clinical studies, that tumors are in fact innervated. So you can see here in pancreatic cancer some big nerves, in breast cancer some bundles of nerve fibers, and in colorectal and bladder cancer some small nerve fibers here and here. And interestingly, it has been shown that the high axon density is correlated with aggressive tumor and shorter survival. And also they did some experiments where they removed these innervations, and it causes some decrease of tumor growth and decreased metastasis. So all of this means that the tumor are innervated by a newly infiltrated axon that exerts a pro-tumoral activity. But however, the opinion diverged on the meaning of the presence of the nerve structures. Because are there persistence of pre-existing nerve structures that is embedded in the tumor, or is it some newly formed nerve structures? And also, is there axonal plasticity and spotting at the nerve endings? So to answer to these questions, we choose to look at the pancreatic cancer. So more especially the pancreatic ductal adenocarcinoma, which is called PDAC, and it's a tumor of exocrine pancreas. And there is an increase of the incidence, and there is no effective therapy for this cancer. And also, you have some axon guidance alteration in this cancer. And as I've shown previously, it has been shown that there is some nerves inside the tumor, and it has been proposed that the tumor are able to form some new nerves. So the pancreas is innervated by all the different system, so of the peripheral nervous system. So you have the sympathetic neurons in green, the parasympathetic in blue, the sensory in red, and the enteric system. And here I will only show you the results that I have with the sympathetic system that can be very easily immunolabeled with the tyrosine hydroxylase, which is an enzyme that is involved in the synthesis of the noradrenaline. So here you can see it's the 2D sections of a piece of the pancreatic lobule with the TH staining. And in fact, we wanted to look at the whole structure of the network to see some plasticity. And in the 2D section, it was very difficult to see such differences during the tumor development. And what we wanted is to have a 3D imaging of the whole neuronal network to be able to quantify more precisely the differences. And so that's why we did some tissue clearing and light sheet microscopy. So here you have the schematic overview of the main steps of the protocol, where first you have the fixation and the permeabilizations, and you have the immunostaining on the whole organ. And then you do some clearing. Here I use iDisco clearing protocol, where you have a first step of dehydratations and the final step of clearing, where you put the sample in the clearing solutions. Here is DBE. So it works to a lot of different organs, such, for example, here I took the brain, and here you see a fixed pancreas. And after all this process, here you have a cleared and immunolabeled pancreas. And so for the imaging, we took the ultra microscope from Miltenyi Biotech. So this microscope, you have here the objective lens, the chamber here where you put your sample. And your sample, in fact, is illuminated by both sides with a very thin light sheet. And the light sheet will take optical sections, step by step, of 2D images. And after, we will reconstruct all the optical sections that we have to have the 3D reconstructions of our tissue. So you can see here the 3D reconstructions of the sympathetic innervation in the 3D pancreatic lobule, where here it's much more easy to look at the whole neuronal network to see some difference of plasticity. So we used the mouse model to mimic the pancreatic tumor, which is called the KIC mouse line. And in this model, you have the normal acinar cells that upon KRAS mutations, these normal cells will they differentiate into epithelial cells or duct-like cells, and will lead to precancerous lesions, which is called ADM. It can evolve after 2 weeks to panning lesions, some other precancerous lesions. And if you had other genetical events, such as the loss of the cancer suppressor gene INK4A, you will have the cancer lesion, so the PDAC. And during the whole process, you have an increasement of the fibrosis. So with our light sheet, we were able to identify all the different pretumoral and tumoral lesions in the pancreas. So here you have a wild-type pancreas lobules, and here you have a KIC pancreas lobule. And you have 3 different stainings. So the topo 3 staining is a nuclear staining. You have in a green CK19, which is stained ductal epithelial cells, and it's also highly expressed in tumors here. And finally, in red, you have alpha SMA, which label vascular smooth muscle cells, and also activated fibroblasts inside the tumor. So we have our 3D structure, and we can come back to 2D that we had during the acquisition with the ultra microscope. And in fact, we found that with the autofluorescence of the tissue, it gives us enough information to identify our structure of interest, such as ADM, pannier, or PDAC. You can see here the normal lobular in the pancreas, and here the pretumoral lesions. So all the data that I will show you, I identify the pretumoral and tumoral lesions with the autofluorescence of the tissue. So here you see the sympathetic innervation of the whole pancreas. So we see that there is a kind of specific pattern of innervation. First, you have here a big branch that will innervate. Here is the spleen that goes through all the pancreas. And here you have two other branches that divide, and there we innervate the whole organ. So next, you can see here the sympathetic innervation in the KAC mice. So you have here in red the tumor, and we can see that inside the tumor, the axonal network is quite disturbed. But we can still find the different branching and the patterning that we saw previously in the wild-type pancreas. So if we look more closely, we see here the KAC and the control. Again, here in red, the tumor. And in yellow here, you have the nerves that are inside the tumor. And we can, in fact, identify the same nerves in the control here and here, here and here. So it means that the nerves that we find here inside the tumor is not some newly formed nerves like it has been proposed, but some pre-existing nerves that are embedded inside the tumor. So there's no newly formed nerves, but we then wonder if there is some axonal sprouting and plasticity. And we know that if one see the axonal plasticity, we have to change the scale, and so to zoom in inside the pancreas. So here it's a zoom of lobule with again the TH staining of a normal pancreas. And if we look now, a pancreas with in the KC mice, so with a tumor, we see that we see there and there two hotspots of innervation. And so when we go through our two dissections and look at the autofluorescence, we see that these hotspots of innervations corresponds to some pretumoral lesions. So we next wanted to look at these innervations at the different stages of the tumor. So we combined the TH staining with the MICA32 staining, which labeled blood vessels, because we know that blood vessels and axons interact with each other, and we wanted to look at the behavior of this interaction during tumor development. So here is the original image, and we quantify and analyze our images with Imaris software. So with Imaris, we use a tool which is called filament tracer that allow us semi-automatically to reconstruct the whole neuronal network, and it gives us some morphological description of the axonal network, such as the axonal density, the branching density, the branch length, the straightness, and the interaction of axons with each other. And we also reconstruct the whole blood vessels in order to use another tool which is called surface contact extensions that use also MATLAB. And you see in yellow here all the contact between the axons and the blood vessels. And again, it gives us some morphological descriptions of these contacts, such as the size, the fragmentation, or the density. So here you can see the sympathetic reconstructions in control asymptomatic tissue, nodular fibrosis, ADM panning, which are pretumoral lesions, and PDAC, so tumoral lesions. And you have in blue the duct-like cavities, which is basically empty space that we removed at our final analysis for the quantification. And so it is quite striking to see that in the pretumoral lesions, you have an increasement of the sympathetic axons density, and it tends to diminish in the PDAC stages. So next, if we look at the blood vessels, In fact, when we quantified the density of the blood vessels, we didn't see any differences between the control asymptomatic, the pretumoral, and the tumoral lesions. But it is striking to see that you have some morphological differences between the different stages. And finally, if we look at the axon and blood vessels contact, you see that in control and asymptomatic, so you see in yellow all the contact, that they are closely in contact with each other. But when you progress during the cancer, you see that these contacts tend to diminish and to be more fragmented. So here is the summary of all the variables that we looked at for the axonal network and the axon blood vessel interactions in the KAC mice for the asymptomatic nodular fibrosis, pretumoral lesion, and tumoral lesions. And as I show you, we see that we have an increasement of the axonal density in pretumoral lesion, but not in PDAC stages. An increasement of the branching and diminution of the branch length as it is newly formed axons. And regarding the contact, you see that you have a decrease of the contact of blood vessels and axons and an increasement of the fragmentations and the density. So then we use all these variables and we did a principal component analysis combined with hierarchical clustering, which is an unsupervised task. And we end up with 3 different groups. So healthy tissue, tumor, and pretumoral lesions. So it means that we have a pattern of innervations that can distinguish the early normal tissue early lesions and PDAC. So this specific pattern of innervation could be a predictive value to the tumor stages. So finally, I will just show you very briefly the work of Jérémy Guillot, a PhD student in the lab, that look at the function of these newly formed axons inside the tumors. So he did pharmacological denervations. So he injected a drug, 6-OHDA. So this drug will lead to neuronal death of only the sympathetic system. So he injected the drug at around 3 and 4 weeks, so before the first pretumoral lesions appears. And he looked first at the survival of the mice. And it was very interesting to see that, in fact, you remove the sympathetic system of the mice, they have a shorter survival. And then when he looked at the size of the tumor, he saw that when we remove the sympathetic system, you have a bigger tumor than the control mice, which is the KAC with no drugs. And finally, he looked at the metastasis in the liver here. And again, you have more metastasis in the treated mice than in the control mice. So he also did this same analysis with a chirurgical denervation, and he has exactly the same results. So it means here that sympathetic fibers has anti-tumoral activity here, and they limit tumor growth and metastasis. So to conclude, I've shown you that you have an axonal remodeling during PDAC development that is developed during pre-cancerous lesions and diminished during PDAC. And you have a specific pattern of innervation that can distinguish normal tissue early lesions and PDAC that could be predictive values. And finally, these fibers has anti-tumoral activity, and they can limit tumor growth and the metastasis in pancreatic cancer. So I'd like to thank all the lab, and more especially Jérémy Guillot, that I work a lot with, and Angélique Puget, a very talented student that I had at the end of my postdoc, and all the other lab members, and again, Miltenyi Biotech for this opportunity to show my data. And thanks to you for your attention. Thank you very much for your presentation. And now it's my pleasure to introduce the next speaker of BraYn Educational Symposium. She is Daniela Carnevale from Becton Dickinson, and she will present a symposium entitled Neuroregulation of Immunity in Cardiovascular Disease. I leave the word to Daniela Carnevale.
Okay, as introduced, I will talk about the role of neural control of immune response involved in hypertension. Hypertension is a complex condition that derives from the imbalance of several pathophysiological systems that normally contribute to blood pressure regulation. And as shown in this picture, we can see that the brain, the autonomic nervous system, kidney, and vasculature typically regulates blood pressure and keep blood pressure levels normal. However, one or more than this system is dysregulated, the blood pressure can rise and give origin to the condition known as hypertension. We have currently a variety of antihypertensive therapies that typically target this system. And although many therapies are available and patients can have quite control of the blood pressure level, there are still patients that have resistant hypertension, and especially that continue to show target organ damage despite the antihypertensive therapies. In fact, it is well known then that when blood pressure is constantly high, target organs of high blood pressure like the brain, the heart, kidney are continuously damaged. And in terms of innovation in this sense, we know that in the last decade, we assisted to the discovery of a new mechanism, a new pathway that participates to blood pressure regulation and is related to the immune response. In this recent review published in Nature of Immunology, we have a nice timeline of the discoveries of papers that link immunity and hypertension. And although there are several old observations that associate a dysregulation of the immune response with the onset of hypertension, only about 15 years ago, we assisted to the first work, mechanistic work, that clearly showed that the immune response participates to hypertension. In this work, the group of David Harrison exploited the model of RAG1 knockout mice that are basically devoid of T and B cells to challenge them with a typical model of experimental hypertension that is obtained by the chronic infusion of the peptide angiotensin II. With this model, during some weeks, typically 2 or 4 weeks, we assist to a typical rise of blood pressure that reflects hypertension. And in RAG knockout mice, we can see that these mice are protected, meaning that lymphocytes are necessary for hypertension. Some years later, the same group also published another interesting work that also clarified that the mechanism of co-stimulation of T cells is particularly important for blood pressure increase. In fact, with pharmacological means and genetic approaches, they inhibited the co-stimulation axis of B7, CD86, and they demonstrated that by these ways, mice were protected from hypertension. In addition, there was another important work, because this work, published in 2010, demonstrated for the first time that while angiotensin II in mice is usually administered in the periphery through osmotic mini pumps, the effect is mediated by the brain. We know that some specific brain regions, the brain regions that are more are lined by a leaky blood-brain barrier, the circumventricular organs, are densely enriched with angiotensin receptor. And in this paper, the authors demonstrated that when we lesion this area of the circumventricular organs and we infuse angiotensin II in the periphery, we have a significant protection from blood pressure increase, as shown here, meaning that hypertension is induced when angiotensin II is sensed in the brain. But the other interesting thing that in this paper was shown was that the immune activation typically observed in this model of hypertension was impaired as well. In fact, the typical tract that is observed in angiotensin II treated mice is the activation of lymphocytes that are recruited to target organs like the vasculature and kidney, and by ablating the circumventricular organ's response to angiotensin II, they obtained a protection from infiltration of T cells in target organs. And our group was interested in understanding where these cells that infiltrate the aorta or other vascular districts and the kidney are coming from. And in order to answer this question, we decided to generate a particular mouse model that is obtained by spleen transplantation. And basically, we used the CD45.1 and 2 leukocyte antigen to transplant the spleen of CD45.1 into a mouse with a C57BL6J background. And by this way, if we infuse angiotensin II in these mice and then we perform flow cytometry analysis in target organs like the aorta and kidney, we can distinguish between cells coming from the spleen and cells coming from the other part of the body. And as shown here, we demonstrated that in kidney aorta, we can find cells that are clearly of splenic origin, meaning that the brain senses angiotensin II and the spleen in some way is able to respond to this challenge. Also, we splenectomized mice and we infused angiotensin II in order to understand whether these cells are really relevant for hypertension. And by measuring blood pressure with TACAF, we obtained a result that clearly demonstrated that the spleen is important for blood pressure increase in hypertension, as shown here. But the question was how the brain can be connected to the spleen. We know that the spleen is innervated by a sympathetic nerve, that is the splenic nerve, that typically takes origin in the celiac ganglion, that is connected through the spinal cord to the brain. However, the work done by the Kevin Tracy group also demonstrated that there is another level of regulation of this pathway and is obtained by an afferent pathway of the vagus nerve that can connect on the celiac ganglion and modulate splenic immune responses. Thus, we decided to understand whether this pathway is recruited in hypertension, and we established a system, a miniaturized system for microneurography of the splenic nerve. So basically, with this approach of recording directly the activity of the splenic nerve, we measured the firing of the splenic nerve in mice infused with angiotensin II. And as shown here, we can see that these mice typically increase the firing of the splenic nerve in terms of firing frequency and amplitude gain. More recently, we have also approached directly the celiac efferent of the vagus nerve, and we developed another system to record the vagus nerve activity that is basically different from the splenic nerve, that is sympathetic. In this case, we can appreciate that the burst activity is different in terms of spikes, and we, with a MATLAB analysis, we were able to count the burst of mice with angiotensin II, finding that this is significantly higher than the other mice. In addition, in order to directly prove that this connection is really established at the level of the celiac ganglion, in collaboration with the group of Kevin Tracy, we obtained another kind of information that is here represented. Basically, what we did was to inject with a tracer, with a virus that reports a tracer in the vagus nerve nucleus in the brain. And on the other side, we also injected a retrograde virus in the spleen to analyze in the celiac ganglion the connection between these two nerve fibers. And as shown here, you can see that there are a co-staining of the efferent vagus nerve here and the nuclei that give origin to the splenic nerve here. In the double staining, you can appreciate the direct connection in the celiac ganglion. But what about the possibility to directly recruit the activity of the splenic nerve through the vagus nerve? In order to do this, we also established another system of bioelectronic modulation of this pathway. Basically, this is a schematic representation of the experiment, and we can see that in a sham animal, we have the recording at the level of the splenic nerve here. That is the same kind of analysis that I have showed before. And in mice that are stimulated at the level of the vagus nerve, we have here the protocol. So basically, the information that we can get from this experiment is that if we stimulate the vagus nerve celiac efferent, we can evoke the sympathetic nerve activity at the level of the splenic nerve and noradrenaline release inside the spleen. That is something that mimics what happens with the infusion of angiotensin II. But what about the mechanism inside the spleen that is recruited by this sympathetic nerve activation? We investigated this pathway in a work published some years ago, whereby we were able to demonstrate that there is a particular growth factor that is released inside the spleen when sympathetic nerve is recruited, in this case by angiotensin II. This is one of the key experiments that showed that this growth factor, that is called the placenta growth factor and belongs to the family of VEGF growth factors, is released inside the spleen. Here we can see that angiotensin II induces the activation of this growth factor in the spleen, and that in mice that have a denervation of the splenic nerve performed by celiac ganglionectomy here are unable to express PLGF inside the spleen. And more interestingly, we also challenged these mice with angiotensin II, PLGF knockout mice that are all knockout mice, and we found that these mice are protected from hypertension. But more importantly, when we exploited again the generation of the spleen transplanted mice, thus having a knockout spleen in a wild-type mouse or just the opposite, and we infused angiotensin II, we measured blood pressure, finding that it is sufficient to have a knockout of PLGF inside the spleen to be protected from hypertension, independently from the presence of PLGF in the rest of the body, thus indicating that PLGF upon a neuroimmune activation inside the spleen is able to promote an immune response that is necessary for blood pressure increase. Thus, summarizing this first part of the data, we can see that angiotensin II activates the brain, that through a vagus splenic connection releases noradrenaline in the spleen to promote PLGF expression. But what happens inside the spleen when the PLGF is released? PLGF typically acts through a canonical pathway that is called VEGF receptor 1. The VEGF receptor 1 belongs to the family of the VEGF receptor that typically transduce the signal of the VEGF family. However, PLGF has also another co-receptor or receptor that is less known for its function, that is called neuropilin 1. The first thing that we did to investigate this pathway was to investigate the expression of these two receptors inside the spleen in basal condition and upon angiotensin II activation. And as shown here, you can see that VEGF receptor 1 and neuropilin 1 are expressed basally in vehicle mice and are upregulated upon angiotensin II. The expression is not completely overlapping because VEGF receptor 1 characterizes more the vasculature, but both of them are expressed in the marginal zone of the spleen, where we know that PLGF is released as well. Thus, to investigate the role of VEGF receptor 1, since the knockout of this model is embryonic lethal, we exploited a model that has a tyrosine kinase defective domain. So basically, in this mouse model, we have the receptor, but the receptor is unable to transduce the signal. We infused angiotensin II in these mice, and to our surprise, we had no effect because these mice were hypertensive like wild-type mice, suggesting that the canonical pathway that transduces PLGF signaling is not relevant for this neuroimmune-mediated hypertensive response. And thus we decided to investigate more neuropilin 1, that is known as a co-receptor, is more important for axon guidance in neurons and is less known for the function of VEGF growth factor family. An interesting work, however, of some years ago demonstrated in a system of immune cells isolated from humans, an in vitro system, that in the process of maturation from monocytes toward the dendritic cells, neuropilin-1 is upregulated, indicating that besides its function of axon guidance, it can also be important for promoting an immune response. And this is interesting because angiotensin II has been demonstrated to promote this process of maturation of monocytes-derived dendritic cells. And in particular, in the model of angiotensin II-induced hypertension, it was shown that in the spleen, there is a significant proportion of dendritic cells that derive from monocytes, and that these cells express the co-stimulation markers that are important for priming T cells that in turn are recruited toward the target organs. Thus, we questioned about the role of this process of maturation from monocytes toward dendritic cells, and with a system whereby we can stimulate and reproduce in vitro the process of maturation, we analyzed the effect of placenta growth factor that was added to this system. And by characterizing the response by immunocytochemistry, we were able to appreciate that the addition of recombinant PLGF was able to induce a state of maturation of dendritic cells that was enhanced as compared to a standard protocol. More important, when we analyzed this response by flow cytometry, we were able to quantify this phenomenon and to really characterize the expression of the co-stimulation molecule CD86 in response to PLGF and compared to standard condition of maturation. And this allowed us to conclude that PLGF is important in the process of maturation of dendritic cells from monocytes in vitro. What about in vivo? In vivo, we challenged mice with angiotensin II, and we analyzed the same fraction of monocytes-derived dendritic cells that is shown here. But in order to connect this with the signaling of PLGF, we measured by flow cytometry the expression of neuropilin-1 in this fraction of cells. And as shown here, angiotensin II increases the expression of neuropilin-1 in the fraction of monocytes-derived dendritic cells. And more important, when we denervate the spleen by celiac ganglionectomy, this effect is significantly blocked, suggesting that the axis PLGF neuropilin-1 is recruited by angiotensin II to promote the immune response. And in order to take information on the relevance of this pathway in vivo, we generated a mouse model with an ablation of neuropilin-1 in the myeloid lineage in order to inhibit this signaling in the population of cells that give rise to the dendritic monocytes-derived dendritic cells. This is the schematic of the mouse model generated, and this is the control experiment just to demonstrate that the inhibition of neuropilin-1 was selective in the fraction of monocytes of the myeloid lineage. That is the fraction that then we subject to the protocol of stimulation with the standard medium maturation and the addition of PLGF. And what we observed is that in the absence of neuropilin-1, PLGF is unable to promote the maturation of dendritic cells, and also at the level of quantitative analysis by flow cytometry, we can appreciate the same results. This indicates that the pathway responding to PLGF inside the spleen is transduced by neuropilin-1 inside myeloid cells. And again, if we go back to the in vivo model and we use the mouse model generated by blocking neuropilin-1 in the myeloid lineage and then infusing angiotensin II, we can investigate what happens at the level of maturation of dendritic cells, monocytes-derived dendritic cells. As shown here, we can see that there is a significantly reduced maturation of dendritic cells, monocytes derived dendritic cells, and in particular, the interesting thing is that these fractional cells significantly block, have a significantly hampered expression of the co-stimulation molecule that is necessary for priming of T cells. And how this can influence the process of blood pressure increase. It has been shown by our work and other group experiments that when we infuse angiotensin II in mice, the priming of an immune response inside the spleen promotes the aggression of T cells. And this is an important and crucial moment of the immune response involved in hypertension, because the T cells egressing from the spleen are in turn recruited in the target vasculature, where they really can contribute to blood pressure increase. As shown here, if we measure the content of T cells by immunohistochemistry in the spleen of mice infused with angiotensin II, we have a typical reduction. This reduction is inhibited in mice that are defective on neuropilin-1 in the myeloid lineage. The same effect is also observed when we perform a quantitative analysis by flow cytometry inside the spleen of mice infused with angiotensin II in the presence of neuropilin-1 in the myeloid lineage or in the absence of neuropilin-1 in the myeloid lineage. Basically, we can appreciate a significant reduction of both CD4 and CD8 T cells, but especially relevant is the reduction of CD8 T cell effector T cells, and the fact that neuropilin-1 inhibition in myeloid lineage completely block this phenomenon. And this is important because we have observed that the CD8 T cells are in turn recruited into target organs like the kidney, where their presence is crucial for altering the normal mechanism that regulates blood pressure. In fact, if we look at the kidney of these mice and we analyze the infiltration of the T cells in the target vasculature of the kidney, we can see that normally this is significantly increased, the content of CD8 T cells. In mice with neuropilin-1 knockout in myeloid lineage, this effect is inhibited. And the same effect was observed by analyzing the quantitative data by flow cytometry. And in addition, since we have seen that CD8 T cells infiltrating target organs participate to damage of tissue, we also analyzed by staining the fibrosis in kidney and by analyzing the alterations at the glomerular level. We can see that the typical damage, renal damage, that is observed in hypertensive models induced by angiotensin II are significantly reduced in mice that have neuropilin-1 knockout in myeloid lineage, meaning that the inhibition of recruitment of the CD8 T cells also reflects in protection from target organ damage. But more importantly, these mice, when challenged with angiotensin II and measured in blood pressure with the tail cuff or with the telemetry, are protected from the typical increase of blood pressure as shown here in the quantitative data or in this representative recording. This is a typical response, hypertensive response to angiotensin II. Mice that have neuropilin-1 knockout in myeloid lineage are protected from this effect. So to conclude and summarize these results, we have seen that angiotensin II activates the brain and promote a vagus nerve splenic nerve connection realized at the level of ciliary ganglion, and this releases PLGF through a noradrenaline mediated release inside the spleen. And at the level of the spleen, the release of PLGF is necessary to promote the maturation of monocytes derived dendritic cells that in turn are able to promote a mature CD8 T cells activation that, when infiltrate the target vasculature, are able to effectively affect the mechanism regulating blood pressure and then induce hypertension. And with this, I have concluded. I would like to thank all the people of our group that contributed to this work. I would like to thank all the granting sources of this work, and I would like to thank BD for the opportunity to show you and share with you this data. And thank you for the attention.
Thank you for your nice presentation. And just to remind to all of you, the correct affiliation of Professor Carnevale is IRCS Neuromed, University of Sapienza, Roma, and she's an invited speaker from Becton Dickinson. Okay, sorry for misunderstanding. And now I leave the word to the chair of Neurophysiology and Neuroplasticity session.
Okay, let's go on. The next speaker is Gabriele Nardi from Scuola Normale Superiore of Pisa, and we'll talk about modeling focal cortical dysplasia by driving PTEN gene deletion with a novel Cre-amplifying reporter. Please, Gabriele.
Yes, thank you. Hi, everyone. And I'm sharing my screen. I hope you see it. Isn't it? Okay, so hi everyone, and thank you again for giving me this opportunity. I'm Gabriele Nardi, a PhD student of Scuola Normale Superiore and in Pisa, and today, as a member of Gian Michele Ratto team, I'm going to show you our last effort, which is going to be published soon. In the first part of this presentation, I'm going to bore you a bit talking about genetic mosaics and why we needed to realize a specific tool, which we call Beatrix, to model them. But then we will go through the results we obtained from its in vivo application to model a human pathology, that is focal cortical dysplasia. But let's start with the question, why mosaics? Well, indeed, when we think about a monogenetic disease of the central nervous system, we imagine a germline mutation shared by all the cells of an individual. However, somatic mutations can happen during development, leading to the coexistence of mutated and healthy cells in an individual, or in this case, in the brain. Now, it is interesting that in the past few years, somatic mutations have been associated with brain diseases leading to autism and epilepsy. But it's even more interesting that in some pathologies, patients show a phenotype only if there is such a mosaic condition. I mean that all homozygous mutants are healthy. Now, you might understand that in this field, being able to model genetic mosaicism is important, and we really wanted to model mosaic mutations. But how could we do it? How could we obtain a mouse brain with both healthy and mutated cells also being able to finally tune the proportion between these two populations? It was not easy. Indeed, the most common approach to model gene deletion is to start from a cell line or a mouse strain in which a gene of interest is fluxed and use the Cre recombinase together with a fluorescent reporter like this one that changes color upon Cre recombination, let's say with the aim of reporting the excision of the target gene. However, this is good enough if, for example, you transfect your cells with the reporter alone, of course obtaining only, in this case, red and wild-type cells, or if you co-transfect your reporter with, let's say, a huge amount of Cre recombinase, obtaining only green knockout cells. But with the low amount of Cre that is needed to model a mosaic, we got a lot of these yellow cells here due to weak or transient Cre activity. Now, these yellow cells are beautiful, but tell us nothing about the state of our gene of interest. This is why we developed Beatrix, a plasmid that includes a Cre gene that is expressed upon Cre recombination, providing, you see, a positive feedback that promotes the recombination of the other copies of the plasmid in the cell, leading to Cre amplification. And this amplification can turn this situation of ambiguity into a situation of full recombination, in which a cell goes from yellow to green, and all the sequences, including the gene of interest, are recombined. So we compared the performance of these two systems in vitro by counting the number of non-red cells as a function of the proportion between Cre and reporter used for the transfection, as we did before. And we saw a tenfold increase in sensitivity of Cre of Beatrix compared to the non-amplifying system. And most importantly, the number of yellow cells was significantly reduced. So we were happy. We moved in vivo, transfecting neurons with in utero electroporation and looking at them with 2-photon imaging. We obtained these wonderful mosaics, and we confirmed that Beatrix performs better than non-amplifying reporters. And in the end, we obtained this plot here that shows that we have been able to realize what we were talking about at the beginning. So we can also tune our mosaics, ranging from 0 to 100% of green cells, that mean knockout cells, among the transfected one, simply changing the proportion of Cre and Beatrix plasmids used for the electroporation, and all of this keeping the number of yellow cells down to negligible values. So at this point, we were ready to use Beatrix on a floxed mouse strain, and we choose to target the PTEN gene. PTEN is a phosphatase that regulates and inhibits the mTOR pathway. Needless to say, this regulation of this pathway can be deleterious for many cell lines, and neurons are not an exception. Indeed, there is, let's say, an entire family of pathologies that are called, indeed, mTORpathies that can affect the nervous system, and one of them is focal-cortical dysplasia. The affected children show epileptic phenotype and intellectual disability. Moreover, their neurons, their knockout neurons, their mutated neurons, show abnormal size and migration defects. Indeed, when we transfected PTEN floxed mice with Beatrix and Cre in utero, we obtained these cortical mosaics of red, so wild-type cells, and green, so PTEN knockout cells, which actually recapitulates this phenotype. And moreover, in agreement with the pathology and with other, with previous observations, we also reported this increase in dendritic spine density that can lead to an excess of excitatory inputs in the knockout neurons, which is not, I mean, surprising considering that we're talking about a pathology with epilepsy. Okay, this is the moment at which a physiologist would say, okay, let's look at the electrical activity of these brains, because can the mutated neurons that in the end, I mean, represent a small fraction of the world population induce alteration in the overall cortical activity, as it is in the human disease? So to this purpose, we recorded local field potential from mice, both in the injected hemisphere and in the contralateral hemisphere, under urethral anesthesia. In this condition, such as in deep sleep stages, the cortex oscillates between two states, one state of activity called up state and a state of relative silence called down state. And it does so with a high degree of spatial synchrony. I mean that if you record the local field potential from the same area of the two hemispheres, you obtain two, let's say, almost identical traces. However, this is not the case with our mice. We saw two main kinds of loss of synchrony. This one that I show here, in which upstates from the mosaic hemisphere last longer, and this other one in which the amplitude of the upstates is reduced. We also quantified this loss of synchrony looking at the average cross correlation calculated on time window centered on the upstates. So to briefly explain this plot, please think that the height of this central peak here indicates how much two time series are correlated, and this width here indicates for how long they are correlated, okay? But as you can see clearly, there is a general reduction in the cross correlation of the two hemispheres in mosaic mice. So slow wave oscillation in our mosaic region is affected, but is the contralateral hemisphere affected as well? This is an interesting question, because if it is so, it means that the pathological effect of the mutation is not confined to the small area of the mosaic itself. And so, since slow wave oscillations occur, let's say, in a frequency range that goes from 0.5 to 4 Hertz, that is called the delta band, We measure delta band power both in the contralateral hemisphere of our mosaic mice as well as in the control mice. And interestingly, we realize that mosaic does affect the contralateral hemisphere, as you can see here from a reduction in its delta band power. Moreover, we observed two different kinds of epileptic-like events. This kind of spontaneous bursts during the downstate, and this kind of rhythmic activity that actually resembles the subtle EEG alteration observed in human patients. So to conclude, we have seen that in the end, with Beatrix, we accomplished our goal of, let's say, modeling a mosaic mutation of a human brain disease. However, to fully exploit the potential of this tool, we are going to look at the effect of changing the number of knockout neurons, so tuning the mosaics, in order to tell apart cell-autonomous effects from network effects and to correlate the severity of the phenotype with the number of knockout neurons. And we are also going to combine Beatrix with a genetically encoded calcium sensor in order to image the activity of both population of healthy and mutated neurons at the same time and in vivo. So I thank all my lab team, and I thank you for the attention. I hope you enjoyed.
The first question that I received is, if I'm not wrong, the grasp reconstitution is irreversible. Thus, how could you distinguish the GFP signals of the stable synapses from that of the non-stable one?
So the synaptic grasp basically works if there is a structural connectivity and they are functionally active. So as he suggested, like I won't be able to see the grasp signal if there is no more connections available. So the condition is it should be structurally connected and it should be functionally active.
Okay. Okay, I see. And another question is, did you apply your synaptic approach to a pathological context like a synaptopathy or epilepsy?
Now we are like in the validation stage, and we have validated in the in vitro, and now we are validating in the in vivo. So if the system works, like we are sure if the system works in the in vivo, then we will be using it in mouse models for like degenerative diseases or other diseases.
Okay, thank you.
Yeah, but we haven't done it yet.
Thank you. So I would switch to the questions for the educational symposium. So Pellegrino. Or perhaps if there are problems, I can continue with.
Okay, there is a question for from the audience for Daniela Carnevale.
Who will read? Yeah, Daniela Carnevale is on.
Is on?
Yes, I'm here.
Yeah.
Okay, awesome. Apart the level of CD86, have you tested the functional effect of PGF on monocyte-derived dendritic cell like cytokinase secretion and or capability of induced T-cell proliferation?
This is an interesting question because what we are doing now is something that is ongoing, is to investigate how the process, the promotion of maturation of dendritic cells can affect the function of T cells. One of the possibilities that they, through cytokines or chemokines, they affect the mobility and thus the aggression, and the other is, for example, that they affect the rate of proliferation. We have just preliminary data on this because we have tested the effect on proliferation, and for now it seems that there is no strong effect on that. So we can speculate that the effect is mainly related to the capability of induced aggression, so chemotaxis of these T cells, but this is still in progress.
Okay. Simona, do you have other questions for? No? Okay, so perhaps I can continue with the other two speakers, and in particular with Gabriele Nardi. So the first question I received is, okay, hi Gabriele.
Hi.
So the first question is, the phenotype you see is the result of in utero electroporation. Would you expect similar effects by inducing PTEN somatic mutation later in life, or do you need to hit a critical plasticity time window?
Well, there are other models in literature in which the mutation is induced with the injection of viral vectors of Cre later after birth. And there are similar phenotypic effects. For example, the enlargement of the cell size increase and increase in spines. You see also if you induce the mutation later. However, with that kind of injection, you also obtain much more mutated cells. So, for example, in those works in which you also see an electrophysiological phenotype, this might be due to the presence of many more cells that are mutated, and especially there is a work that also links the number of mutated cells in the hippocampus. So, and the degree of, let's say, delamination in the hippocampus with the electrical abnormalities, in this case, epileptic events. So we think that by inducing this mutation during development with in utero electroporation, we also cause a set of networking if we impair the networking of the cortex.
Thank you. And another question is, can you explain the differences and the advantages of your Beatrix approach compared to other systems like the MADAM system in producing mosaicism?
Yeah, well, there are other systems, but these other systems are usually, there are different, let's say, different specificities of the other systems. For example, MADAM has a lower Cre sensitivity because its sensitivity is the same as, for example, a standard ROSA 26 locus. And also there are false positives and false negatives that depends on the Cre promoter that is used with that system. And for example, for MADAM, there are other systems, for example, that have different, let's say, I mean, different, that are different. For example, they use more systems, not only Cre, but also Flip and also Teton. For example, there is a system called Supernova. So we think that Beatrix is a very easy system, and it does not require the generation of a mouse line, and can be used for in this specific for modeling this specific developmental diseases.
Thank you, thank you. So we can proceed with the BraYn educational workshop. The chairman will be Gabriele Sansevero.
Hi everyone, and welcome to the educational workshop. I would like to introduce you Vincenzo Di Ruocco from Beckman Coulter that will hold a presentation entitled Innovation in Flow Cytometry evolution of the Cytoflex platform.
Okay, thanks for the kind introduction. I will share my screen.
Yes.
Yes.
Okay, so first of all, thanks the organizers for giving me the opportunity today to have a talk during such interesting meeting. I'm Vincenzo Di Rocco, I'm manager in Beckman Coulter, and I'm responsible for flow cytometry application support in South Europe. And today I will talk you around Cytoflex and how this instrument can fit in neuroscience researches. So this is my agenda. As I said, I will talk to you about the technology first, and mainly about flexibility and easy to use, which makes this instrument very easy to adopt. So we'll talk not about conventional application like CD4, CD8, but really specifically, I will give you some examples how flow cytometry can apply it in neurosciences. And then just the last slides about the roadmap, so the evolution of this platform, and then resources. And then the team in Beckman Coulter then is able to provide you support. So this is the Cytoflex family. This is the first instrument that was launched in 2015, so 5 years ago. As you can see from the picture, it's a very small one, but is very powerful because it can goes up to 3 lasers from 4 to 13 colors. So it's pretty actually, yeah, flexible thing, even in the configurations. The following year, we launched on the market the Cytoflex S, up to 4 laser. So we increased the flexibility, adding yellow laser, near UV laser, in order to expand the possibilities. And then the biggest brother in the family is the Cytoflex LX, which can work up to 6 laser, 19 or 21 colors. So a lot of really markers to detect our cells and classify our cells. So why this instrument is so innovative? Actually, if we can imagine to open the lid of the instrument, what we will find are these blocks. And these blocks are what we call WDM, which are the optical bench plus the detectors. So here, you can see here, this is the fiber optic, which brings the light, the detected light from our cells, from our fluorochromes, and we need to actually split our light into the optical bench. And you can see here everything is miniaturized. These are the bandpass filters, and behind the bandpass filter there are these detectors which are APD, so really different from the last generation of instrument which are based by PMT. So again, the detector's function is still the same. So what we want to do is transform the light into electrical signal, but with this new technology, we can have a much better quantum efficiency. Okay, so you can see here this picture that clearly show you the better sensitivity that we can have with this technology. So there are some advantages using the Cytoflex and this technology. Some of them are about sensitivity, some other are about spreading. When we, for instance, we define multicolor panels and we are using a lot of fluorochromes together to better understand, identify our cells. And then from easy of use perspective, we have linear response. I don't have time enough to discuss a lot about these advantages. I will focus more about the application, but give me just a couple of slides to show you this. So these are rainbow beads. These are commonly used to understand and compare instrument qualities, and the separation that we can have among the first peaks across these aids give you the idea about the sensitivity and the resolutions. So as far as you can see, you can clearly see clear separations, and this will enable researcher to really differentiate small differences among what is negative and what is really dimly positive. Okay, and how can we get this? Actually, these are the settings, and they came from the QC. So the first thing in the day, we launch the QC, we check the instrument, we also get some recommended settings that we can use for the application. So it's very, very easy to work with this instrument. And then I was talking to you about linear response. So this is what happens with the PMT, with the old generation. So I cannot predict actually when I change my voltage in my my detector what can happen about MFI, about the intensity of that channel. As opposite with in the APD situation, when we change the voltage applied, we can still understand or forecast what will happen next with the intensity of the light. So since we are talking about a lot of fluorochromes, a lot of colors together, and there are interactions that is called compensation, and this is the trickiest point for flow cytometrists. The system itself automatically calculate the compensation, but more of this, if we need to change for some reason the voltage applied because we want to highlight some specific small population, really dim population, okay, we can increase, for instance, the voltage applied to that specific channel, and still my compensation will be automatically readjust because we can the system can use some algorithms internally to automatically do this. Indeed, when we import compensation, the system itself will ask you if you want to import the matrix and convert based on the current gains. Okay, so yeah, I want to give you the flavor or the idea that this is a very easy to use and very user-friendly instrument, but still with very high performances. But it's flexible because Cytoflex, I mean, we keep flexibility such important, such seriously that we put in the name of the instrument itself. So we show an image about the footprint. So it is very small, and we can for sure transport this in any lab. We can also use in some researcher using into some boats for doing some marine biology, just to let you know that the easy of transportability. We can put this in a biohood. We have multiple configurations. The upgrade is a software, so pretty instant. There are different costs according to the configurations, of course. The detection size particle can be very different. We can work with very small particle down to 80 nanometers, so very, very small world, up to very big 30 micron cells or particles. We can also customize our filters. We can work in a microplate, not only in a single tube. We can have different software from a default, very simple, basic, up to electronic record management in case you want to comply with CFR 21, this kind of compliance, okay? Especially important for industries and biotech. And this actually, all these characteristics, taking all together, actually enable what we call flow democratization. So now I want to go into the neurosciences, and I pick some application notes or white papers out of our collections, and I will describe shortly some of them. You can go to our website and you can educate yourself, or you can download this document. They are free, for free. And the interesting thing is that, of course, there are results inside, but there are also protocol and detailed protocol how to get such results as real papers. So in this case, what I'm showing here is identification and characterization of BraYn tumor initiating cells using the Cytoflex. So of course, we need to mechanically disaggregate these cells, okay, with scissor and scalpels. Then we need to use some enzyme to get them into suspension. Then we need to culture them in serum-free natural media. And then actually there are a lot of steps, but they are all described in protocol. I cannot go in here into the details. But what we can actually see is a classical flow cytometry pattern. So we have our cells. We can use some viability dyes in order to get what the cells that are really live. And now in this case, we are using 4 or 5 markers to classify these cells. Okay. Another example is this one. This is a little bit different, and it's not focused on the sensibility or resolution, but more or on the peristaltic pump. Okay. In this case, we are studying a neurotransmitter receptor. And what we are doing is recording physiological changes in response to an agonist. Okay, so what you see here is the fluorophore behavior. This is one acquisition. And so I see my cells with this emission in this channel. And then I can put some ATP in my tube. And then I see the spike of my cells, and then after kind of 50 seconds, they go back to their steady state. And this is possible because there is no compressor, and my tube is, you see here, the tube is actually external to the system. So I can constantly mix my cells, or in this case, I can drop the agonist directly into the tube while the system is acquiring the cells. So that's why actually I see this shift in fluorescence in really a few seconds. And this is a pretty unique feature for a Cytoflex. Then, of course, we can have other applications. So cycle is pretty common application, but we can apply this to our human brain tumor stem cells. And again, into the white paper, there is a very nice detailed procedure how to get it and all the reagents required. So here are my cells, singlets, and these are classical cell cycle picture that we can get from our software Kaluza, analysis software Kaluza. Another example I want to give you today is about detection of the extracellular vesicles. Yesterday, Beckman Coulter sponsored a very nice talk. I think this is a recording. I hope you can get back to this. So Dottoressa Claudia Maria Radu from Università di Padova, she talked about extracellular vesicle as emerging biomarkers for diagnosis and therapies. And what is interesting here is that the violet side scatter channel actually really enable to better perform this kind of application. As you can see here, this is the smallest size of my beads, and this is 100 nanometers, and these beads can be clearly differentiated by the noise in this case. So we had a lot of success so far with Cytoflex. We are very happy about this instrument. This is the install base globally and in Italy. So there are a lot of instruments already in the market. We can provide the instrument itself. We can also provide liquid handler to prepare samples. We can also provide reagents, okay? So antibodies to stain our cells. They can be in a liquid format or in a dry format. And we also have all the software to analyze and continue the experiment with analysis. So Kaluza is a very user-friendly software, and it matches very nicely with the Cytobank, which is more sophisticated software, cloud-based, which enables researchers with unsupervised gating strategy and gating algorithms like BSNI and others. So what about the future? Actually, this is not future, this is already present, because this year we launch the twin brother of the Cytoflex. This is the DXFlex, the same technology, but with the IVD, CE IVD mark. So we are selling this into hospitals for clinical analysis, and we already have 50 units in Europe. For researcher, next year, early next year, first quarter next year, we will launch a Cytoflex sorter. So the same technology, no peristaltic pump, but different fluidic system, but still same APD, same detectors, and same user software interface. In this case, we are not just analyzing our cells, but we are really separating, and we are able to get back our cells from our sample. I will give this presentation to the organizers, so you can go back to our website, or if you're interested in COVID-19 research, or you can get back to our collection of application notes that I showed you before. I need to thank my team. So Angela, Jorge, Alice, Tommaso, Letizia, we are providing support all around Italy. Jorge is in the team, the person who is more focused on research. And during the lockdown, he also had Cytoflex in his home to better provide support to our customers. But all of us can use Bachmann Connect as a virtual remote support to our customer in a safe and virtual mode. So I ended my presentation and ready to get some questions in case. Thanks for your attention.
And thank you very much, Vincenzo, for your interesting presentation. Unfortunately, we are in late, so questions should be asked and answered directly in the chat. And we have to move on. For the audience, poster session is now open and the presenting authors are waiting for you in the virtual rooms. And please note that every session will last 1 hour and 20 minutes. The Congress will start again at 2:30 p.m. in the main platform, so see you later.